Bismillahirrahmanirrahim. My dear student of first year, now please I would like to continue my lecture on the cell organelle. Today is the most important organelle of a cell controlling all the activity is the nucleus. Nucleus is a specialized structure occurring in most of the cell except some bacteria and blue-green algae. In human can be given an example very important that they are deceased without nucleus. So it is separated from the rest of the cell cytoplasm by a double layer of the nuclear membrane. This membrane seems to be continuous with the endoplasmic reticulum of a cell. Previously I have told you about this endoplasmic reticulum cell and has pore complex which probably permit the entrance of the large molecule. The nucleus controls and regulates the activity of a cell, growth and metabolism, and carries the gene structure that contain the hereditary information. So nucleus is the master organelle in a cell. So in controlling all the activity of the cell. So it is the important organelle containing the structure which is coming in the next slide. Now, nucleus consists of the gene, the DNA structure that contain the hereditary material in the form of a chromatin material, chromatin or chromosome. Now you all know that the gene is the triplet codon, which is the message for a cell, or is the message of Allah Almighty. It is the message of Kun from Allah Almighty, through which the whole universe is controlling through genes. So it is the gene which is the carrying all that active information for the corresponding cell. Next is the nucleoli, are small bodies often seen within a nucleus. So these are the site for the ribosomal RNA activity. The next is the third gel-like material in which the nuclear components are suspended is called the nucleoplasm. Now this is the diagram which will show you the nuclei present in the three cell corresponding neighboring cell and closed by a cell membrane as well. Now what is the nuclear membrane? The nuclear membrane consists of two lipid bilayer membrane, an inner nuclear membrane and outer nuclear membrane. So this is same as that of the mitochondria, but the membranes are as that of the plasma membrane, that is lipid bilayer. The proteins are sandwiched at the fluid mosaic model you know very well. The space between the two membranes is called the perinuclear space. It is usually about 20 to 40 nanometer wide, this space. The outer nuclear membrane is continuous with the endoplasmic reticulum, that is the part of system of tubular network starting from the plasma membrane up to the nuclear membrane. So it is a continuous channel through which the cell trafficking takes place. The nuclear envelope has many nuclear pores that allow material to move between the cytosol and the nucleus. Now, no structure can pass through the nuclear membrane bilayer, lipid bilayer, but only the way is the nuclear pore which give way, or it is a gateway for the propagation of molecular movement from the nucleus to the cytoplasm and from the cytoplasm to the nucleus. Inside the inner nuclear membrane, you see a intermediate filament protein called the lamins. The lamins is the woven network of intermediate filament which anchor the membrane to the nucleoplasm and give the support of the nucleus. Now next we move on to the slide, that is the nuclear pore complex. This is the diagram which will tell you the structural presentation of the bilaminar pattern, double layer, outer and inner. Now look at this is the outer and this is the inner and this is the perinuclear space, and this one is the nuclear pore complex. In this complex you see there is the well-known pattern of the a channel through which these structures pass in and out from the nucleus. It consists of the outer cytosolic fibrils you see in this diagram. Now, this is the cytosolic side fibrils, and this one is the nuclear fibrils. So it is the two type of fibril you see, that is the inner fibril toward the nuclear membrane and the outer fibrils toward the cytoplasm, and between this is the a nuclear pore having the three types of rings of protein, that is cytoplasmic ring, luminal subunit, and which is also called as annular subunit. And this one is the nuclear fibril, and this is the annular subunit over here visible. Now this is the nuclear fibrils and from the basket-like pattern. So it is the basket-like pattern which is visible in the nuclear basket. Now this is the diagrammatic presentation of a nuclear pore. Now this nuclear pore is the gateway through which all the molecules comes in and out. Now अगर हम बात करें प्रेजेंट सिनेरियो की जो कोविड-19 है, तो the RNA of the RNA virus that pass through this gateway. Now this through gateway pass inside the nucleus. So this is the hypothetical diagram to remind you and to show you the structure of a nuclear pore complex. Now look at the picture, that is the nuclear pore. These are the pictures, diagrammatic presentation of a electron microscopic picture of a nuclear pore. Now these are the three pictures over here, 1, 2 and 3, and this is the hypothetical diagram previously described to you, nuclear pore and the nuclear membrane. Now this is the site. You look at the picture, that this is the nuclear pore area, and this is the inner and outer nuclear membrane, and this is the perinuclear space. Now look at the picture over here. This is again a electron microscope picture of a nuclear pore complex. Now how you see the nuclear trafficking? This is again an example because it is very much important in the today's scenario. अगर आप इसके आज के मौजूदा हालात में देखते हैं तो how nuclear trafficking takes place, what are the structure pass, how the structure pass from inside to outside. It is quite simple, as in the plasma membrane previously described to you. Small molecules are able to pass rapidly through open channel in the nuclear pore complex by passive diffusion. Quite obvious. Passive diffusion means according to the concentration gradient from the cytoplasm to the nucleus and from the nucleus to the cytoplasm. But some of the molecules which are larger and they had to transport it in and out against the concentration gradient and as required. So this needs the energy, and this energy dependent mechanism is for larger molecule, and they are concerned for the protein and the various types of RNA which passes through with the help of energy dependent molecule. In this picture you see that is the proteins and RNAs, they are passing through a nuclear pore with the help of energy mechanism, but the small molecule through the passive diffusion, and this diagram is pictured over here. So this mechanism is very simple diffusion and energy dependent, but through the pore complex system. Now we move on to the very important structure present within the nucleus, that is the chromatin material. This slide will depict the 23 pair of chromosome. It is a male template of a chromosome, 22 autosome, and one is the pair is the sex chromosome having Y chromosome present in this diagram. You see it is the male chromatin slide. So 22 pair that is autosome, and one is the sex chromosome. In male it is XY and female it is the XX. Description. Now, it is the chromatin material is a complex of macromolecule found in the cell consisting of DNA, protein, RNA. DNA, protein and RNA. The three structures are present in the chromatin material. Remember, DNA, protein and RNA. The DNA, which includes genes that are actively transcribed, turned on, is more loosely packed and associated with RNA polymerases referred to as euchromatin. Now, my dear student, यहाँ पे बात समझने वाली है कि very commonly asked in viva examination and in later on your clinical examination, what is the difference between a euchromatin and heterochromatin? Always remember that the euchromatin is the part of DNA which has to be transcribed. The part of DNA which has to be transcribed, उसको रीड करने के लिए उसको नेकेड होना ज़रूरी है. तो नेकेड DNA ताकि उसको टेम्पलेट बन सकें विद द हेल्प ऑफ RNA पॉलीमरेज वहाँ अटैच होगा, उसको हम कहेंगे यूक्रोमाटिन. इट इज़ लेस स्टेन्ड. इट इज़ लेस स्टेन्ड, ये भी याद रखना है, रिलेटिवली लेस स्टेन्ड. दिस क्रोमाटिन इज कॉल्ड एज ए यूक्रोमाटिन. ये बात ज़रूर याद रखें कि ये वो पार्ट होता है DNA का जो कि ट्रांसक्राइब होना है, जिसके आगे ट्रांसक्रिप्ट्स बनने हैं ताकि प्रोटीन सिंथेसिस हो सके. बट द DNA विच कोड्स इनएक्टिव जीन टर्न्ड ऑफ, तो वो पार्ट ऑफ द DNA विच इज नॉट रिक्वायर्ड फॉर द जीन एक्टिवेशन और जीन सीक्वेंसिंग, उसकी ट्रांसक्रिप्शन नहीं होगी. इट विल बी कॉल्ड एज टर्न्ड ऑफ एंड इज मोर कंडेंस associated with structure protein. It is known as heterochromatin. So heterochromatin is the chromatin usually seen during cell division. सेल डिवीजन के दरमियान जो हेटेरोक्रोमाटिन कंडेंस हो जाता है, कोई एक्टिविटी सेल की नहीं हो रही सिवाय इसके डिवाइड हुआ होता है. ये मैंने एग्जांपल बता दी. और भी एग्जांपल्स होती हैं कि एक DNA मॉलिक्यूल कुछ जगह पे हेटेरोक्रोमाटिन है, कुछ जगह पे यूक्रोमाटिन है, और यूक्रोमाटिन जो है याद रखें लेस स्टेन्ड है, और वहाँ पे RNA पॉलीमरेज जो है वो एक्टिवेटेड करता है और उसके ट्रांसक्रिप्ट बनते हैं फॉर द रिक्वायर्ड प्रोटीन सिंथेसिस. माय डियर स्टूडेंट, ये आप लोगों ने ज़हन में बना लेना है यूक्रोमाटिन हेटेरोक्रोमाटिन का डिफरेंस. व्हाट इज द न्यूक्लियोसोम? दिस इज द स्ट्रक्चरल यूनिट ऑफ ए DNA. दिस इज द स्ट्रक्चरल यूनिट ऑफ ए क्रोमाटिन मटीरियल, नॉट DNA. क्रोमोसोम का स्ट्रक्चर यूनिट जो है इट इज कॉल्ड एज ए nucleosome. Consists of a 146 base pairs of a DNA wrap around 8 histone molecules. Now this is आप कंसर्न करें, इसको रिमाइंड करें कि consist of 146 base pair of DNA wrap around the 8 histone molecule. This is called as a nucleosome. DNA look like a string of beads. DNA look like a strings of beads. Beads are the 80 stones, where strings are the DNA which is wrapped around the stone in between the stone beads. So this is the beaded ring appearance. Ring on nucleosome are organized into a large coiled loop held together by a non-stone scaffolding protein that is called as a nucleosome. 8 histone wrapped by a 146 base pair of a DNA molecule around the nucleus, around the histone protein. This is called as a nucleosome. So that is why this nucleosome, when present in a chromosome, chromatin material, it is called as a bead on string appearance. Nucleosomes are organized into large coiled loop held together by a non-stone protein are also present, which is called as scaffolding protein, जो उसको wrap करके रखती हैं. This is the coiling phenomena. This diagram will tell you how the chromatin material is coiled itself. This is the 2 nanometer diameter. These are the two rungs. In between are these are nucleotide, and these are the two rungs. Is 2 nanometer coiling. Coiling means twisting of a DNA molecule. Now over here you see this is the bead on sting appearance. 8 histone molecules and wrap around it the DNA. It is a nucleosome. Histone 8 molecule and 146 base pair of DNA, that is will form the bead, and rest of the DNA is called linker DNA. So in between the two nucleosome you see a linker DNA over here which links between the two nucleosome. So this coiling is when it is coiled itself, the nucleosome come closer. So this is called as 10 nanometer in diameter. Now it is the nucleosome is 10 nanometer in diameter. 2 nanometer in diameter is the actual width, and then become coiling itself come closer, then it is called as 10 nanometer fibers. Now when tight helical formation 3 nanometer diameter, so it will form supercoiling over here starts, and this supercoiling about 300 nanometer diameter will be the supercoiling, and this supercoiling is seen in the metaphase chromosome 700 nanometer तक भी हो सकता है ड्यूरिंग सेल डिवीजन। ये बात याद रखनी है कि सेल डिवीजन में कोई भी जो क्रोमेटिन मटेरियल है वो ट्रांसक्राइब नहीं होता, उसलिए वो सुपर काइलिंग में चला जाता है ड्यूरिंग मेटाफेज, जहाँ पे इक्वेटोरियल प्लेट पे ड्यूरिंग सेल डिवीजन क्रोमोसोम जमा होते हैं, जहाँ पे सेंट्रोमियर के साथ स्पिंडल लगते हैं, तो वहाँ पे 7 नैनोमीटर का सुपर काइलिंग फिनोमेना होता है मेटाफेज में। सो दिस काइलिंग प्रोसेस इज आल्सो ए नॉर्मल फिनोमेना इन दी chromatin material. Now what is the nucleosome? Now this is the diagrammatic presentation of a nucleosome. Now look at the picture. These are the 8 molecules of the histone protein: H2A, H2B, H3, H3, H4. This is the double protein H2A 2 molecule, H2B 2 molecule, H32 and H4. So it is the 8. This is the linker DNA. This is the over here. This is linker DNA, link between the two nucleosome. So nucleosome had 8 histone and 146 base pair of DNA. So these double pair, it is the 146 base pair of DNA, which is form the bead on sting appearance. Now look at the picture again. This is the diagrammatic presentation over there. The wrapping DNA molecule 146 base pair, histone molecule, and this whole structure is called as a unit structure nucleosome in a chromatin material. Role of histone. Now histone molecule is concerned for the transcription mechanism, whether the histone will pack itself to form the more coiling phenomena, no transcription. When transcription activities, there is a molecular organization. So over here, look at the picture. So the histone molecules are quite tightly packed, and there is no area for gene transcription. So this is the coiling phenomena. The methyl group is most abundant in this picture. The histone tail and the genes are hidden within the histone molecule and not give way to the transcription factors to attach on this. Tightly packed together, transcription factor cannot bind the DNA and genes are not expressed. जब जगह ही नहीं होगी, न्यूक्लियोसोम जो है इतने टाइट हो जाएंगे कि वो जगह ही नहीं देंगे ताकि ट्रांसक्रिप्शन फैक्टर जो है वो अटैच होकर ट्रांसक्रिप्शन को स्टार्ट कर सकें. So this phenomena, the role is basic histone molecules, histone protein. Now below diagram, it will tell you that this is the opening up, elongation, the histone tail will become away, and histone acylation result in loose packing. When methylation, then acylation. When methylation occur, it is tightly packed. When acylation occur, it will become loosely arranged. Now there is the area. Now this is the look at it, this is the gene area. Now this gene is now bare up. It is open access for transcription factor. Now these transcription factor जो है, this is concerned for DNA accessible and gene will be active site and transcription will be takes place because there is a wide gap. So this acylation and methylation phenomena, the key role is the stone. When methylation will take place, the coiling phenomena will occur. When the acylation will take place, the gapping phenomena, the nucleosome become away. And when this is away, so what will happen? There will be translation of this naked DNA for transcription factor, and hence transcription will be take place. Now this is quite obvious to you, and you know very well that is the double helix Watson-Crick DNA model, the rung, the sugar phosphate, handrail, and adenine paired with the thymine. Look at picture. GC pair with the adenine with the thymine, and guanine with the cytosine. You all know that is the 4 nucleotide: adenine, guanine, cytosine, and thymine. And in case of RNA, it is uracil. The pentose phosphate form the handrail. The rung is the internucleotide bond, the covalent bond interaction that will form the rung, and this is the very important Watson-Crick DNA model. You all know, all the students know very well. Now this is the messenger RNA. Now look at the picture of messenger RNA. The cytosine, adenine, uracil, and guanine, cytosine, adenine, and uracil. So these are the nucleotide sequence which we will see in the RNA: adenine, cytosine, guanine. This is the molecular formula: adenine and uracil in place of thymine. Now this is the messenger RNA template. Now these are the nucleotide bases which are now empty to pair in the ribosome to form the protein synthesis. This is the hypothetical picture of a messenger RNA. Now what are the chromosome? This is the macroscopic picture of a chromosome, not in depth in the chemical composition. It is the morphology of a chromosome. It consists of a centromere. It is also called as a P arm, which is above arm. This is the P arm, and this is the Q arm, which is also called as a long arm. The P arm is also called as a short arm. Short arm, Q arm is the long arm. They are united within a center. It is called a centromere. Now, this is the morphological picture of a chromosome. Now, types of chromosome. Now, these are the various types of chromosome: telocentric, acrocentric, submetacentric, and metacentric, according to the location of the centromere. Look at the picture. These are the centromere. So, where it is located, when it is almost in the center, it is metacentric. Where is the subcentric mean? That is the long arm, and the short arm are quite obvious in this picture. Now this is a satellite. This is the satellite mean drumstick appearance. It is called as the acrocentric type of chromosome, whereas there is no short arm, very negligible short arm, but the long arm is prominent. It is called telocentric. These are the morphological characteristic types of the chromosome. What is the nucleolus? It is the largest structure in the nucleus of the eukaryotic cell. It is the site known as the ribosomic biosynthesis. Now nucleolus, you all know, in my previous all the lecture I have described to you. मैंने बताया है कि nucleolus उन सेल में prominent होगा जहाँ protein synthesis ज़्यादा होगी. So, although it is present in all the cell, but it is prominent, well known, and well visible in those cells where the protein synthesis will take ample amount. Nuclei are participating in the formation of signal recognition particle for protein synthesis. Cell response to stress. Nuclei are made up of protein, DNA, and RNA, form around specific chromosomal region called the nucleolar organizing region. Nucleolus is formed in a specialized chromosome which is known as nucleolar organizing region, NORs, on chromosome 13, 14, 15, 21, and 22. These are the number of chromosomes where this nucleolus is meant for to arising from the nucleolar organizing region, which is transcripts present within the nucleolus. is not surrounded by a membrane, is organized around the chromosomal region that contains the genes for the 5.8s, 18s to 28s RNAs. Ribosomal RNA, eukaryotic ribosome contain 4 types of RNA designated to 5s, 5.8, 18s and 28s. So these all are the RNAs which are meant for the ribosomal synthesis, and eukaryotic cell contain 4 types: 5s, 5.8s, 18s and 28s, and these RNA are present in the nucleolus. It is not the membrane bound, but it is present in the specific nucleolar organizing region NORs on the chromosome 13, 14, 15, 21 and 22. Structure is the 3 major components of the nucleolus, that is the fibrillar center FC and the dense fibrillar component and the granular component, DFC, GC and FC component. The transcription of the RNA occur in the fibrillar center, that is the FC. Transcription of the ribosomal DNA will take place in the FC region. The DFC contains the protein fibrinogen, which is important in RNA processing. So इसका मतलब ये होगा कि DFC contain the protein which is the fibrinogen protein, which is meant for the RNA processing. The GC contain the protein nucleophosmin B23 in the external image, which is also involved in ribosomal biosynthesis. So granular protein is the nucleophosmin, which is about B23, and it is involved in the ribosomal synthesis. This is the structure of FC, DFC and GC components of the nucleolus. Now look at the picture. This is the hypothetical diagram. Granular component, dense fibrillar component and fibrillar component. Look at the, so this is granular component, this is nucleolus, and this is the dense fibrillar component, and GC granular component. So these are 3 are the components visible. Corresponding structure will be described in the previous slide. Nuclear matrix is the network of, the last component of the nucleus is the nuclear matrix is the network of fiber found throughout the cell nucleus and is somewhat analogous to the cytoskeleton. The nucleus matrix along with the nuclear lamina aid in organizing genetic information within the cell. Many proteins associated with the matrix have been discovered. Proteins such as scaffold, matrix associated protein have also a role in the organization of the chromosome. So various proteins are also present, and these are scaffold protein, matrix associated protein and chromatin organizing proteins are present within the nuclear plasma, and the nuclear matrix is similar, that is the cytoskeleton, analog of the cytoskeleton you see in the cytoplasm. Now these are the nuclear matrix associated protein. This is the hypothetical diagram, but detailed electron microscope picture will try to depict all these proteins. Now this is the matrix associated protein in this diagram you see over here. 10 nanometer fibril, this is nuclear lamin and outer nuclear membrane. So all this structure, tubular structure. So these are the proteinous network in the nucleoplasm. So this is the hypothetical diagram, but you will have to remember the matrix protein as well as the nuclear membrane as well as the chromatin material protein as well. Thank you very much. Today's lecture, it's all about you. Thank you so much.